There are so many parts to the cell membrane that I decided to make a video just for this topic alone. The cell membrane forms a boundary between the cell and its outside environment. It's also selectively permeable, which means it only allows certain nutrients and molecules to pass in and out of the cell. Keep in mind that you're looking at a cutaway view or a side profile of the cell membrane. The cell membrane is composed of a double layer of phospholipids. A phospholipid is composed of a phosphate head, which is polar, and it's got a tail, which is nonpolar, made of lipids. The cell membrane is made of a double layer of phospholipids, and it's got proteins and carbohydrates that are sticking in and out of it. Now, you have to just keep in mind that you're looking at a side profile or a cutaway view of the cell membrane. So just imagine that the cell membrane is covering this whole entire cell. It's like its own casing. A phospholipid has a few main parts. First, it has a phosphate head, which is polar. And because it's polar, it loves the water. It's hydrophilic. Next, it has a hydrophobic tail, which is made from fatty acids, and these are pointing inwards because they dislike the water. They're hydrophobic. Hydrophobic just means that it fears the water, dislikes the water. I think this picture will give you a better understanding of how the cell membrane forms a barrier around the cell. So you have a phospholipid bilayer, and you have the polar phosphate heads, which are facing the water, and then sandwiched in between them are the fatty acid non-polar tails, which dislike the water. There are a few molecules which are embedded in the cell membrane. The first is cholesterol, which helps strengthen the cell. The next is protein, which acts as a channel that allows things to move in and out of the cell. Last but not least, you have carbohydrates, which act as identification tags, and it allows the cells to communicate with each other. The fluid mosaic model helps describe the structure of the cell membrane. It's not a rigid structure, but rather it has a lot of flexibility, and it kind of just jiggles and it moves around. And it's got proteins and all sorts of molecules that are all embedded throughout. This is known as a fluid mosaic model. Obviously, the cell membrane is almost like a barrier, so the cell has to figure out a way where it can move materials in and out of the cell when it needs to. And one of the first ways that it does this is called passive transport. And passive transport means that the cell does not need to use energy to get stuff in and out of the cell. The materials just move across the cell membrane using the concentration gradient. And how this works is it's just moving from a high concentration to a low concentration. It does this on its own. It's almost like moving downhill. You don't need to invest energy to move stuff across the cell membrane. It just happens on its own. Diffusion is a form of passive transport, and it requires no energy. Think about perfume molecules spreading out on its own. This is known as diffusion, and in diffusion, molecules move from an area of high concentration to an area of low concentration. So the perfume molecules start where there's a lot of perfume molecules all surrounding it, and eventually over time it dissipates, it goes into the next room, it's spreading out. So it's moving from high concentration to low concentration, and that is known as diffusion. Osmosis is basically the diffusion of water. It's a form of passive transport, and notice again in the model that I provided how the water is moving from an area of high concentration to low concentration. It just does it on its own and it requires no energy. This is osmosis, which is passive transport of water. Which direction the water moves during osmosis, whether the water goes in the cell or out of the cell, really just depends on what kind of environment or solution the cell is in. Let's discuss each of the three possibilities, starting with the far left, where the cell is in an isotonic solution. Iso means equal, so the cell is in a solution where it has an equal amount of solutes both inside and out of the cell. You can see how I drew 3 green dots representing the solutes inside the cell, and then 3 green dots on the outside. Because it's in an isotonic solution, water moves equally both inside and out of the cell. The cell remains normal. Now if you turn your attention to the panel that's in the center, we're now looking at a hypotonic solution. Hypo means low, so there's a low concentration of solutes in the solution. The cell contains more solutes than the surrounding solution. So I've represented this with green dots. I drew green dots all over the cell to show that it has a lot of solutes inside. Outside, there's very few green dots. So what happens is the water is going to rush into the cell to try to dilute the extra solutes. So water rushes into the cell and the cell starts to blow. It blows up. This is kind of like if you capture a shark and you put it in a freshwater environment, the shark would bloat up. Let's now turn our attention to the panel that's on the far right. The cell has now been placed in a hypertonic solution. Hyper means above or a lot, so that means this type of solution has a lot of solutes dissolved in it. So I represented this again with the green dots. You can see that the cell in the center barely has any solutes inside. Now compared to the surrounding outside environment, there's a lot of green dots or a lot of solutes that surround it. So what happens is the cell actually loses water to its environment. The water's leaving the cell. So this is kind of like going for a swim or taking a shower, and you look at your hands afterwards, they're all pruny, your fingers are pruny, and that's because the water from your hands is actually left. It got drawn out by the salts in the water. We've talked about osmosis and diffusion, which is a form of passive transport. Passive transport requires no energy, whereas now if you look at the model, active transport is moving materials from low concentration to high concentration. It's like pumping them against the concentration gradient. It's almost like pushing a boulder uphill. It requires effort and a bit of energy to achieve this. And here you can see a side-by-side comparison between active transport and passive transport. In active transport, this is like going uphill. You need energy, and you're pumping materials from low concentration to an area of high concentration. Whereas in passive transport, this is like moving stuff downhill. There's no energy that's required, it just happens on its own, and you're moving from an area of high concentration to low concentration. Passive transport is going downhill, active transport is pushing something uphill. The cell can take in nutrients and water in a much faster and efficient way known as endocytosis. There are two forms of endocytosis. Phagocytosis, which is cell eating, it can take in a large amount, and penocytosis, which means the cell drinks or it takes in a small amount. And how this works is the cell gets close to whatever material that it wants, then the cell membrane kind of folds inwards and it engulfs that material and it just draws it in. On the other hand, if the cell wants to remove something from itself, then it's going to perform exocytosis, which is the reverse of endocytosis. So it packages up whatever it wants to throw out or remove into something called a vesicle. That vesicle fuses with the cell membrane, and then the cell membrane just spits it out, and that's how bad materials leave the cell. Here's a summary of what we learned today about the cell membrane. The cell membrane is a fluid structure made of a phospholipid bilayer, and you have proteins and carbohydrates that are embedded within it. The cell membrane can also move materials in and out of the cell through either passive transport, which requires no energy, active transport, which needs energy because you're pumping something against the concentration gradient, and endocytosis, which means the cell forms a vesicle around the cell membrane and it's drawing materials and nutrients inside the cell. There's also exocytosis, which means the cell removes materials using a vesicle that fuses with the cell membrane and it ejects the unwanted material out. Thanks for watching, and I'll see you next time on Win Biology.